Hello again. This is the third of three videos regarding chapter 2 in your textbook, The Chemistry of Life. All right. Remember, in the last video, we talked about the first two major biological molecules, which are the carbohydrates and the lipids. In this video, we're going to talk about the last two of these biological molecules, the first of which is proteins. Now, proteins are long chains of molecules, and the subunits, the building blocks that make up proteins, are called amino acids. Now, proteins are very, very important for a number of different reasons. They help to support the body. They are hormones. Many hormones are proteins. Enzymes, which help to digest and rearrange other molecules, are proteins. They act in defense of the body. Antibodies, for example, which fight off pathogens, diseases that humans can get, are proteins. They also assist in motion. So most of your muscle tissue is protein, and they function in the transport of other molecules. Now, the most important thing that you can remember about a protein is that their shape is what determines their function. Okay, so it's almost like a key fitting into a lock. All right, the key only opens a certain lock. Right, you can't, for example, use your car key to open up your dorm room. All right, you can't use your dorm room key to unlock your parents' house, and so the shape of the protein is very specific to the job that it's designed to do. And there are literally hundreds of thousands or even millions of different proteins out there. And we'll talk about how we form those proteins in just a minute. But let's start with the basic building blocks that we use to build a protein. And those are called amino acids. All right. Now, there are 20 different amino acids that humans use. Now, only 6 are shown on this screen, so don't be deceived by that. But there are 20 different human amino acids, and there are others that are used by other organisms like plants and different animals. Now, an amino acid is similar in structure to every other amino acid. They all have a central carbon, what we call an amino group, which is a nitrogen and 3 hydrogens. Now remember, the schnapps deal. All right, we have these 6 elements are most important, and you'll notice if you look carefully at these that all of those elements are represented: carbon, hydrogen, nitrogen. All right, the nitrogen in the amino group, oxygen. And this, by the way, is the acid group. So this is why we call it an amino acid. And then I don't see a phosphorus in this particular slide, but there is a sulfur located here. All right. So they have these amino acids have a central carbon, an amino group, an acid group, a hydrogen on top, and then what's shaded in blue in each of these 6 examples is what we call the organic chain, or an R group. Now, it's the R group that gives each amino acid its own identity. So each of the 20 amino acids has a different R group. All right, and they obviously are structurally different, and they have different chemical properties. You can see this one, for example, is non-polar. This one is ionized and polar, and so on. Now, proteins are organized at 4 different levels. The first of those levels is simply the amino acid sequence. Whoops. How are the amino acids arranged? So we start with the amino end, and we've got glycine, proline, threonine, glycine, threonine, glycine, glutamine, serine, and so on. And so the chain of amino acids is built up. And this almost works like the letters in a word. All right, if you take 3 letters and you organize them in a certain form, they give you a certain meaning. But if you take those same 3 letters and you organize them in a different arrangement, you end up with a different word, or even again, a slightly different word. All right, and so in these 3 letters can be arranged to make 3 different words, amino acids, depending upon their order in the chain, give you a different structural protein. The secondary structure is either an alpha helix, which is simply a swirly, almost like a spring, a slinky here, which is simply arranged by the hydrogen bonds that you see existing between different parts of the molecule. Or they can be arranged in a beta pleated sheet, which is kind of like a folding structure. The secondary structure is then folded to form what we call the tertiary structure, shown here. And this forms what we call a polypeptide. All right. And the polypeptides can then be grouped together to form a protein. And that's the 4th level. All right. And in this case, in this example, this is a molecule called hemoglobin, which is a molecule that helps to carry oxygen and what makes your blood the reddish color that it is. A hemoglobin molecule is composed of 4 polypeptides. And if you look carefully at the diagram, you can see them. There's 2 of the dark blue alpha subunits and 2 of the teal colored beta subunits in every hemoglobin molecule. And hemoglobin, of course, is a protein. Now, moving on, nucleic acids are the 4th and final of the 4 organic molecules that we need to discuss. And their subunits are called nucleotides. Okay, so they're nucleotide subunits. And their job is to act as the blueprint for proteins. All right. The molecules themselves include 2 terms, and I'm sure you're familiar at least with the second one, DNA. All right, this is what we call the hereditary material. But your genes, your chromosomes, are actually designed to be the blueprints of the proteins that you produce. And so the differences in each individual and between each species are based on the differences in the nucleic acid chain that we build in order to produce the proteins that we need. All right, so certain animals have different proteins than other animals, which have different proteins than plants. And all of that is based on the DNA structure inside our cells. Excuse me. So nucleic acids are the subunits, and they come together in long chains to form DNA, which is the hereditary material. Now, there are 3 parts of the basic building block called a nucleotide. The first is a sugar, and there are 2 sugars that can be used here: ribose, which is found in RNA, and deoxyribose, which is found in DNA. Both of them are 5-carbon sugars. You can see the 1 sugar, 1 carbon, 2 carbon, 3, 4, and 5. And on the 5th carbon, there is a phosphate molecule. This is a phosphorus with 4 oxygens. All right, that's what a phosphate is. Attached to the 1st carbon is what we call a nitrogen-containing base. And in DNA, there are 4 nitrogen-containing bases: adenine, guanine, cytosine, and thymine. We'll get into the structure of these when we get further into the course and talk about the unit on genetics. But there are 4 different kinds. RNA differs from DNA in that there are no thymine nitrogen-containing bases, but rather the thymine is replaced with what we call a uracil. So DNA has 4 different kinds of nucleotides, and RNA has 4 different kinds of nucleotides. But the only major difference is that thymine in DNA is replaced with uracil in RNA. Okay. These things, by the way, are called bases because they have the ability to lower raise the pH of a solution. Okay, so they're literally acids and bases again. Okay, so what are the major differences between DNA and RNA? Well, structurally, in DNA, the sugar is deoxyribose, whereas in RNA, the sugar is ribose. The nitrogenous bases that are included in DNA are adenine, thymine, cytosine, and guanine, whereas the bases in an RNA molecule are adenine, uracil instead of thymine, cytosine, and guanine. All right, so 3 of the 4 bases are the same between the 2 molecules. The difference is thymine in DNA and uracil in RNA. The major difference in structure, of course, is the fact that DNA molecules are double-stranded molecules and RNA molecules are single-stranded molecules. All right, and we'll get again into this a great deal more when we talk about genetics. All right, for now, we're going to talk mostly about the nutrient molecules, which are the carbohydrates, the lipids, and the proteins. We'll kind of save our discussion of DNA and RNA until we get into genetics later on. Now, one other molecule that I want to mention, even though it's not one of the 4 major biological molecules that we've been talking about, is a molecule called ATP, or adenosine triphosphate. It is composed of an adenosine molecule, which is adenine. All right, this is one of the nitrogenous bases that we find in DNA and RNA, and a ribose sugar, which is obviously the sugar which we find in RNA. But rather than just having one phosphate attached to it, it's got 3 phosphates here. Now, this molecule is important at the cellular level because it's an energy-carrying molecule. The bonds between the phosphates actually are really storage of a great deal of energy. And so when you break the bond between the phosphates here and here, you release a great deal of energy, which can then be used by your cells to carry out all the metabolic processes that they need to do. All right. But I wanted to point it out here because obviously it's very closely related to the RNA molecules and the DNA molecules that we talked about in the previous couple of slides. Okay, so this slide summarizes really what you need to know about the 4 major biological molecules. All right, notice you've got carbohydrates, lipids, proteins, and nucleic acids here. Examples of each: for the carbohydrates, there's monosaccharides, disaccharides, and polysaccharides. For the lipids, we've got the fats, the oils, the phospholipids, and steroids. For proteins, I didn't give you a specific example, but rather more of their uses: structural, enzymatic, carrier, hormonal, and contractile. And nucleic acids are DNA and RNA nucleotides. The monomers are shown here: glucose for the carbohydrates, glycerol and fatty acids for the lipids, amino acids for the proteins, and nucleotides for the nucleic acids. And then their functions are listed in the last column. All right, short-term energy for carbohydrates, long-term energy for lipids, a bunch of different things for proteins: support, metabolism, transport, regulation, motion, and so on. And then finally, nucleic acids are used for the storage of genetic information. Now, the last thing I want to talk about in this presentation is my list of important things. These are things that I'm going to give to you over the course of the first few units that I want you to make a list of. I want you to put this list in a special place in your notebook or on a special piece of paper or something else, because I promise you that from this list of important things are going to come the long answer questions on your final exam. Okay? The first important thing that you are going to be asked about on your final are the building block molecules, all right, which is what this chapter was really, the second half of this chapter anyway, was really all about. I want you to be able to understand what the process of hydrolysis is, as well as the process of dehydration synthesis, right? The taking apart of complex biological molecules, and dehydration synthesis is the building of these complex biological molecules. And the biological molecules themselves, of course, are the carbohydrates, the lipids, proteins, and nucleic acids. These are summarized, of course, in the previous slide. So make sure you have a good look at them. All right. So that, my friends, is the end of this lecture. And so the conclusion of Chapter 2, the chemistry of life. And I hope that you find all of this very helpful.